Hey everybody, my name is Omid. I'm currently a research nephrology fellow at UCSF, and I studied internal medicine residency, studied some immunology back in my days, and I also just love teaching, and I wanted to create starting lecture series that focuses on some critical thinking and some topics that I just think is really interesting. Maybe just through talking about it, some little bit of inspiration for people in different studies and disciplines, but also maybe if they find it useful for their own learning, targeted in like undergrad, medical school, or other like health sciences. So this is specifically focusing on immunology for this particular lecture. And also, it is going to be about B cell development and how an antibody is generated. I just think this is one of the most interesting discoveries that I learned about throughout my schooling over the years, and it all centers around a certain topic, which is pretty cool. So the way the structure of this lecture is that we're going to talk about a clinical question, critical thinking reviews, major players, and the processes, and then also just some immunology things to go over. So starting off, these are the players we're going to most likely talk about throughout the lecture series, not all today, but in general. B cells, T cells, different types of T cells, macrophages, natural killer cells, dendritic cells, neutrophils, eosinophils, and monocytes. So these are how most of them are going to be depicted throughout the pictures. And, if you find it useful, feel free to pause so that you can draw things and think through it on your own as well. This can both be used for you listening to me, but also just reading through my lecture. So basically, we're going to start with a question. So a patient has a mutation, complete absence of a lymphocyte TDT, terminal deoxynucleotide transferase protein. The activity, gone. Okay, what is the most likely consequence? Okay, so we're going to start by discussing, and then we'll come back to this question. So Frederick Bohlem was a scientist in the 1960s who unveiled a non-template DNA editor. So from your schooling, you'll probably remember that DNA polymerase is a very important editor that uses template DNA from one half of your own DNA, right, the double helix, and it creates the other parallel to it. However, what we've discovered for B cells and T cells is that the generation of their receptor is made through non-template DNA editing. So T and B cell antibody receptor generation is done by what's called somatic recombination of separate genes. So what happens is that rather than simply being generated when exposed to an antigen, our body trains our adaptive immunity to not recognize itself and thus create immune responses to non-self antigens, such as bacteria, viruses, and parasites. And this concept is known as tolerance to self, which means no response. So we're going to back up for a second. So the first fascinating concept is that our body does not know what is a pathogen necessarily. It is a little bit more nuanced than that. There are different innate responses and signals that our body can use to recognize bacteria and viruses, technically. But the general concept is that the actual antigen, which is a part of the pathogen that we've mounted a response towards, that part we actually didn't recognize as ourself. Meaning that when the antibody and T cell receptors are made, they are randomly made. And while they're being made, if they recognize you, like for example, protein in your skin or your heart or your lungs, those antibodies and T cells and those cells die, actually. And the only surviving ones are the ones that actually go on to mature and create these receptors for a response. So that concept is called tolerance. And the way they're made is called somatic recombination. So looking here, we're zooming into a B cell, and this is the VDJ constant region segment. This is the DNA that's used to make the antibody. Importantly, this cDNA becomes the constant region of the heavy chain, which is this longer one, and that parallel side on both sides that's attached, that's called the light chain. And VDJ are the segments at the variable region of the heavy chain and light chain. That is what randomly gets assorted with different numbers of each of them that will then, there's different segments. There's 1 through 30 for V, and J has a couple, D has a couple. They're randomly assorted again before expression. So through that somatic recombination with RAG, you have this formation. And then later on, you can have what's called somatic hypermutation to then add in other changes in the lymph node to mature this receptor even more. The protein that does this VDJ recombination is called RAG, and TDT, which comes from our question, happens later when random nucleotides are inserted after these VDJ recombinations occur to then further create diversity. Okay? So now we're going to focus on some B cell development. This is like a classic page that we will discuss, and also this is a classic page that probably will be on your exam. So B cell development happens in the bone marrow. Okay? B cell, bone marrow, stromal cell, they interact. The B cells that recognize self antigens die, and the ones that recognize foreign antigens survive. When they survive and leave the bone marrow, they go to the lymph nodes, spleen, and the gut, which has another set of lymph nodes there, called mucosal associated lymph tissue, or MALT. And so those cells become mature B cells, and then they become a plasma cell and a memory cell, which then are releasing antibodies, specifically the plasma cells. Memory cells become dormant. Moving forward, the B cell lineage. So how does this all happen? Okay, so starting with a stem cell becomes an early pro B cell, okay? That early pro B cell, notice that it has nothing on its surface right now, okay? It does something called heavy chain DJ recombination, okay? Then it becomes a late pro B cell, so heavy VDJ recombination. So you're rearranging the DNA to make the variable region of the heavy chain. Now you have a large pre-B cell. This guy has a receptor which is called a pre-B receptor. It's basically attached with a fake light chain to have this fake antibody almost, or fake B cell receptor, until the small pre-B cell and the immature B cell is where you have light chain formed. So the light chain goes through VJ rearranging or recombination with the RAG protein, and now you have an immature B cell with both VJ and VDJ recombination done. That guy is going to go to the lymph nodes, become matured, and you're good to go. Now, before you do that, there's a self-recognition test. And once it's cleared, this receptor being not self-recognizing, then it will go and successfully leave the bone marrow, okay? And it will express IgM and IgD, okay? Moving on, this is an explanation here of what's happening. Remember, the constant region is what determines the type of antibody you're making. So it's constant mu region for IgM, constant gamma for like IgG, okay? And then this is the D and J segments of the heavy chain DNA. And when you're making your large pre-B cell, which is right here, this is demonstrating where in the DNA we're talking about. So these are the segments of V, D, and J, and where it's basically, this is the DNA still, where it's ultimately, or excuse me, this is the DNA. This is the actual heavy chain, right? And it's combined with a surrogate light chain. So this is all referring to this generation right here, which we referred to earlier. And you'll see that the variable regions, this is where all the magic happens for what it'll recognize, okay? And the immature B cell, which is this guy, right? Again, this is your variable regions. So you have your constant light chain region, constant heavy chain region, and these are your variable parts of your heavy chain and your light chain respectively. Okay, another interesting concept is the idea that how many gene segments in your Ig loci are there? Well, this goes back to above. I just want to show that there's not that many, right? So you have all these variable regions and diversity and joining regions. These are what they ultimately stand for. Not that many. So how do you make more diversity for your B cell receptor and antibodies? That's the TDT. So during the generation, going back up here, of your variable regions, some of this is basically interjected with nucleotide insertion. So as VDJ recombination happens, random nucleotides will also be inserted between segments by the TDT protein. So to answer our question, lack of TDT will lead to lack of antibody diversity. So you'll have increased risk for infections. You can have an immunocompromised or variable immunodeficiency disorder, right? And one more concept to talk about up here is called allelic exclusion. And what this means is that you have two alleles, right, in all of your like DNA, right? You have two sets of chromosomes. So once VDJ recombination occurs for your heavy chain, right, if it's successful, it will turn off the other allele. If it's not successful, it'll use the other allele. And if that one doesn't work, then the whole cell dies. That's called allelic exclusion, okay? And that's part of the heavy chain success. All right. So moving on a little bit, this is going to the T cell receptor. You have alpha and beta chain rearrangements, right? So both of these together are what make your T cell receptor. You have an alpha segment on one side and you have a beta segment. Very similar concept, but ultimately the RAG protein rearranges your VJ for your alpha and your VJ for your beta. And together, that RAG recombination is what leads to your variable regions up here, basically being nice and prepared for recognizing antigens that are presented to them. And we'll talk about that at a different time. And ultimately, the number of human T cell receptor gene segments and the sources of T cell receptor diversity compared with immunoglobulins. So T cells have more up front, but the somatic hypermutation not included in this number. So basically, B cells have more specifying capabilities that happen later during their maturation. And then only half of kappa chains use nucleotides. So not all of these alleles have the same number of kind of variations. But the number is quite a bit, right? 10 to the 18th or 10 to the 13th. It's quite a bit of diversity. So it's a lot of antigens that can be recognized. So overall, thank you for listening. This was basically talking about B cell receptor development and the two very interesting kind of concepts and discoveries, which is one is this RAG recombination, a VDJ recombination. I think it's just a very interesting discovery that ultimately shows how random T cell development, receptor development is, and antibody development. And then secondly, a very fascinating topic, which is related to autoimmunity, is tolerance, okay, or self-recognition. And so we can get into autoimmune stuff a different time. But thank you for listening and hope to see you guys later.